Welcome to Brain Ponderings, your window into the world of neuroscience research. I'm Mark Matson, the host. Today, it's my great pleasure to welcome Maria Ioannou. Did I get it right?
You sure did.
And that's a Greek name?
It is, yeah. That's
Okay.
right.
And actually, I had to look it up. I cheated because I didn't want to embarrass myself. And she's doing some really interesting work on lipids, fats, in membranes of cells in the brain and but elsewhere in cells. And there's all sorts of different lipids and they play all sorts of different important roles. And they're kind of understudied in the areas of, I think it's understudied anyway, the areas of brain aging, neurodegenerative disorders, which is kind of the avenue we're going to talk about. She's Maria's Canadian. She's up in Edmonton, where she's an assistant professor of physiology at the University of Alberta. And she's also the Canada Research Chair in Brain Lipid Biology. Okay, so she's the person to talk with about brain lipids. So Maria, can you talk about your early education and training, and even before that, when did you get interested in science?
Yeah. Um, I went to undergrad I did a general life science degree. And I think, like most students, I thought I would apply to medicine because that's what everybody thinks they're going to do. And that's a very, I guess, prominent career path. And I got into research in my fourth year, actually summer before fourth year. I was in this program that didn't have an honors thesis, but I really wanted to see what it was like or get a feel for it. And I was very fortunate to find who ended up being my mentor at the time, Dr. Margaret Fanstock. She was completely full. She didn't have any spaces for undergrads. She'd already taken 5, which now, as a PI, I realize that is an enormous undertaking. But she said I could come and wash dishes and shadow people. And I think I spent more time in the lab than the people she was paying. And so she said, you know what, I'm going to make space for you. And if it weren't for her, I don't know what I would be doing career wise. And then once I started in the lab, I originally thought, I'll just do this to get a feel for it, and then maybe I'll apply to medicine from here. But then once I started doing experiments, I'm like, okay, I'll just keep doing this until it stops being fun, and it just never stopped being fun. So I just went with it.
That was at McMaster University.
That's right. Yeah.
Then you went to McGill, which is they're very famous in neuroscience. A lot of
Yeah.
history of
History. That's
yeah.
right.
That they have the Montreal Neurological Institute, and
Yeah.
Penfield was there, and it's
Yeah.
uh,
Brenda Milner is still there.
and and Hans Selye, where was he? Do you know Hans Selye, the stress
No, I
pioneer? He was
don't.
up there somewhere.
Yeah.
Yeah. Okay. And then so your PhD work at McGill, whose lab were you in and what was your project?
Yeah. So I worked for Peter McPherson, and I would say Peter was my PhD mentor. Peter is still my mentor. I still talk to him all the time. He still helps me tremendously. It's such a special relationship. Um, when I went to Peter's lab, I was in a neuroscience program. I was in a neuroscience institute. My main focus was membrane trafficking. That's what Peter was studying, endosomal trafficking. I wanted to learn more about that. And my project started at a very fundamental level. We were looking at RAB-GTPases. These are these proteins that regulate membrane transport, and trying to figure out how they're regulated. And there was, I guess, a decision to be made whether or not we try to push this into a neuroscience project, or there was a very sort of easier path that went into cancer biology. And so Peter said, just go with,
An easier path where it's very likely you're going to get data
yeah,
and have a
it
thesis.
just made more sense. And there was just, it was easier
Yeah.
experiments. And so he's more very, very supportive of just like go where the data takes you. Don't try to force it to be something that you think it should be. And so I got a degree in neuroscience, but my first and bigger paper is on epithelial cancer cell migration,
Well,
which was really cool.
there's brain cancers
Yeah.
and a lot of things you're working on doesn't matter so much what cancer
Nope.
cell. Yeah.
Exactly. And so actually that is something we haven't touched the glioblastoma, but it's always on the back of my mind of something I'd like to go back to.
I had a graduate student, the NIH, when I was there, it had a program called the Graduate Partners Program, where a student applies for the program and at a university, and then they have a mentor at a university and a mentor at NIH. So I had a graduate student, Justin Lathia. He got his PhD from Cambridge, but he did the work in my lab. And then he's a full professor at Cleveland Clinic working on glioblastoma. In my lab, in the lab in Cambridge, he studied neural stem cells.
Yeah.
Now he's studying cancer stem cells.
Yep.
Yeah, so it's
So
interesting.
yeah, there's a lot of overlap. So it's
Okay,
definitely
and then you went to this, and I haven't been to this place in Virginia. You went next. I grew up on a farm, and so you went to a farm in Virginia.
I did. So I did my postdoc at Genelia Research Campus, which is the Howard Hughes Medical Institute. And it was extraordinary. I just loved it there. So I guess I loved it there for so many reasons. First, I had two terrific mentors. So as you can tell from what I've been talking about, but I've had great mentors all along the way. So I'm very grateful for that. But when I was at Genelia, I worked for James Liu, who was at the time just starting to be an independent researcher, and Jennifer Lippincott-Schwartz, who was more senior, well established, and it was really fun to have these two amazing people coming at questions from different angles, from different experiences, and both super supportive.
And Jennifer had like a more 30,000-foot view of the field.
Yeah.
And then the junior one that's got to focus on specific problem. But yeah, she was at NIH for a while.
Yep, that's right. So when I started, she was toggling. She hadn't entirely wrapped up her lab at NIH, and she had started at Genelia. So she was wrapping up one. We went to her retirement party at NIH, and I remember thinking, one, the traffic. Oh my God, I don't miss the traffic. But then also the security to get in to go for a seminar session was a lot. But I understand why they have that in place. But I appreciate my entry into the institute here, where I just
so this happens by coincidence. The previous person I did a podcast with is named Nick Betley.
Okay.
And he was at Genelia too.
Oh yeah.
We did a lot of. So they're really good at imaging.
Yes.
And developing new technologies.
Yes.
And then coupling imaging to electrophysiology or whatever. So this is where you got interested and focused on the lipids, are really nitty gritty.
That's right.
So can you start by like just describing what are some of the key lipids in the membranes of neurons and other cells?
Sure. So major lipid classes would be, we've got triglycerides. These are sort of the storage type of storage lipid when the cells need to sort of pack it away to use later. Phospholipids are an important one that we study. They make a lot of the membranes, the plasma membrane, as well as all of the organelles are made out of these phospholipid membranes. Cholesterol, which is very important in terms of ApoE and disease in the brain. It's of course very important in the periphery as well. This is one that we're interested in, but we don't study as directly. maybe one day. We're starting to a little bit. And then one lipid class that we've recently got into a little more with a postdoc that started about 3 years ago is sphingolipids. So these are very complex types of lipids that they're also on the membranes, they act as signaling molecules, and they do a lot of things as well. And so I would say those are the major ones that at least we think about.
Okay, and okay, so there's so much to talk about, but I'd
Yeah.
like to focus on a few of your studies that are really important advances, and they both have implications for brain aging, Alzheimer's disease, and other neurodegenerative disorders. And so you had well, I don't think this was part of your postdoc. I don't know. Okay, so maybe first describe
Yeah.
what your key project in your postdoc. I don't want to completely skip your
Oh yeah. So the postdoc project was looking at really the transport of lipids from neurons to glia. So when neurons get really stressed out, they have these excess lipids. Most cell types will just store them in lipid droplets, but neurons, especially in vivo, so in the brain, you can make them form them in culture, but in vivo, they don't like to do this as much. And so as a strategy, they ship them out and they get taken up by neighboring glial cells. So different
and
other.
and they do this because the lipids could potentially damage the neurons, or why?
Yeah. So we've since we alluded to it in that paper as well as some of the work by Hugo Bellin, who is also really central to that discovery. And then we built on it since. So these lipids are oxidized. That means that there are these reactive oxygen species that damage a lot of macromolecules, and they damage the lipids. So these lipids are highly reactive, and if this gets out of control, then this can kill the neurons, and the neurons are highly susceptible to this type of cell death, which is called ferroptosis. Yeah, so
And
by
so,
shipping these damaged lipids out, it
so
protects
this
them.
is a paper in Cell in 2019.
Yeah,
And so how did you go about, uh, so you actually looked at
yeah,
these lipid containing vesicles, and you looked at their movement.
yeah,
And how did you do that? You've got to follow them.
yeah. So that was where imaging came in, because the background to do those experiments came from Hugo's paper where he did Drosophila genetics, and he would just stress out the neurons and then see these lipid droplets appear in glia. But we wanted to watch it happen. Um, and that also allowed us to be able to probe mechanisms a little deeper. And so what we did was we took I called it in the lab, I call it a sandwich assay, but it's essentially you take one cover slip and you plate neurons on it. You take another cover slip and plate another cell type. So in this case, they were astrocytes. And then you load the neurons with a fluorescently tagged fatty acid. You sandwich them together, but they're separated by little balls of paraffin wax. So the two cell types can't physically interact. And then you can look for the appearance of the fats that started in the neurons that then go into the astrocytes.
And
And
are
it's
these
really,
fatty
yeah.
acids, they're smaller than triglycerides and diglycerides, or?
So the fatty acid of those original studies, it's called Red C12, and it's it's a fatty acid, but it can be metabolized by the neuron. So it can get incorporated into triglycerides, or it could get incorporated
I see.
into phospholipids. So in those original experiments, we couldn't tell what the identity of it was while it was being transferred, right?
I
We
see.
just knew
Yeah.
that it was being transferred
Yeah.
and it was still in a lipid form. But at the end of that paper, we didn't quite know what the lipid was.
And so the mechanism, the machinery, I guess, the molecular machinery for the movement of these lipid-containing vesicles, outside, I guess I assume, so do they fuse with the outer membrane of the neuron and then release the fats, or does the whole vesicle containing the fats be released?
So so this is what we sort of focused on when I started the lab. It's like in the postdoc, we said, okay, there's this transport process. If you block it, it's very bad for the neurons. So then when I started the lab in Alberta, the main questions was how does it transfer? What's transferring? What are the mechanisms of transfer? And I think that there are multiple ways that fats can go from one cell to the other. So the first way that we found was a process called lysosomal exocytosis. So in the cell, you have lysosomes, and they're the degradative organelles. So when the cell is stressed, it will induce autophagy. All that will end up in the lysosome, and
So,
then it
so
will
that's one thing I remember from my high school biology. Mr. Welty was a really good teacher, and we're going over like kind of the basics of what's organelles,
yeah.
but he would call the lysosomes the suicide sacs of the cell.
They're like garbage cans,
Yeah.
although if you go to a lysosome conference now, that's sort of like the running joke that they're the introduction of the garbage cans because they do so much more
And
in terms of,
recycling.
yeah, and metabolism,
Yeah.
especially like macromolecules and solutes and etc.
So the
Uh,
entire so the lysosome releases its contents outside
it
the cell.
just spits it all out. Yeah. But in order to trigger that, it takes a lot of oxidative stress. And so we're not entirely sure if this is like a last ditch attempt for the neuron to save itself. And that maybe that's not used at physiological states. Maybe this is only like
But what
later.
about the time course? It could, what about, I mean, I'm assuming your experiments are fairly short-term
Yeah,
exposure to
yeah,
stress rather than weeks or months or years.
that's right.
Yeah.
Yeah,
So
yeah,
it could occur, it might get to some tipping point,
yeah,
like the climate change is going on now,
yeah.
and
And
when
then,
we get to a tipping point, and we're all going to be wiped out.
yeah.
So there's a tipping point. Okay.
So that was one way. The other way is through the ApoE lipoprotein particles. And so that was the paper that we had that just came out.
Uh,
I guess the final version just came out a few weeks ago. But you have these lipoprotein particles that are largely released by astrocytes, though microglia they're ApoE positive, and I'm sure we'll talk about ApoE in a minute. But they're major lipid transporters in the body and in the brain. They can dock on neurons and sort of suck out all of the damaged lipids directly from the plasma membrane. So those are the oxidized lipids that they can remove, and that also protects the neurons from cell death. And I think that happens. I think that's more of a physiological protective mechanism that's happening all the time. Yeah.
so you say you have ways to block the removal of the oxidized lipids,
Yeah.
and then the neurons are
They'll die.
dying and
Oh yeah.
degenerating. Do you have the opposite? Do you have a way to enhance the removal of the oxidized lipids?
Yeah, so what we to block it, we pinpointed one of the transporters. It's a transporter called ABCA7, and it is a risk factor for Alzheimer's disease. So that was quite interesting to us because it puts all of these proteins on the same path. If you knock out ABCA7, the lipoproteins can't efflux, or so they can't suck the damaged lipids out anymore, and then the
So
cells die.
there's people with a mutation or variant of that AB,
CA7.
uh, yeah,
Yeah.
that they have increased risk for Alzheimer's.
Yeah. Same with ABCA1, but we were very surprised that we knocked out ABCA1 as well, and we didn't see any effect. So we were quite
Is
surprised by that.
that transporter expressed just in the brain, just in neurons, all over, all cells?
Uh, it shouldn't just be in the brain, no.
Okay.
And I
And then
don't think it's just in
And
neurons
we'll
either.
talk about apolipoprotein E,
Yeah.
and which I haven't heard much about this transporter, so it must not be as quantitatively big a risk factor as
No, that's right. It
apolipo.
just came out in some more recent genetic screens looking at more subtle risk factors. There was a paper also by Hugo Bellin's group. The first author was Matt Moulton. So he looked at these GWAS risk factors and linked a whole bunch of them along the way of this transport pathway. So there's
Ah,
efflux from the neurons, so the receptors that, or the, sorry, transporters that take it out, ApoE, which we'll talk about, and then the receptors on the astrocyte side that pull it up. And all along the way, mutations in many of these steps all sort of lead to the same phenotype, which is neurodegeneration.
okay, interesting. Right,
Yeah,
but the way you can enhance the so you would the
yeah,
idea would be to enhance the
transport.
activity or
Yeah,
amount of the transporter.
yeah. So one way that we can or in the paper, the way we probed it was just by adding exogenous lipoprotein particles. So if you just add more lipoprotein
Ah,
particles, you can efflux more of the lipids, and then that protects the neurons. But you need that receptor to be there. If you knock it out, it won't work.
and are these lipoproteins similar? Well, they're similar to. They're not LDL, HDL.
No, they're very, very simple versions. So LDL in the brain, there's one lipoprotein particle. They call it HDL-like. It's very small, so it more looks like LDL in the periphery, but it's really high in ApoE. And um, yeah, when we wanted to model it, that also has a lot of different lipids in it. So it's got phospholipids, it's got cholesterol, it's got some sphingolipids, etc. Less neutral lipids. In our model system, it's very simple. It's just one phospholipid and ApoE. So it's essentially a liposome. You've got
Ah,
POP-C, so phosphatidylcholine, which is the most abundant phospholipid, and ApoE. But that allows us to
I see.
mix and match the ApoE to sort of play.
Okay, that's neat. All right, so let's talk about. Did we talk? Yeah, I don't know if you ever came across Alan Butterfield. Um, he did a lot of work. Actually, he was at the University of Kentucky uh, when I was there, and on on membrane lipids and particularly oxidative damage to membrane lipids and Alzheimer's. And so, well, let me give some background on that because so when we were there, he had some evidence that, and we collaborated with him that so in Alzheimer's disease, you get the amyloid beta peptide. It's aggregating, self-aggregating, forming plaques,
Yep.
and it can be toxic if you expose neurons to aggregating amyloid beta peptide. It can be toxic. We found, and we never, I don't think we ever published it, that the aggregating amyloid beta peptide seems to accumulate in patches on the membrane of the neurons. And those patches correspond to what are called lipid rafts,
Huh. Yeah.
which are rich in cholesterol and sphingolipids. And then, anyway, if exposed neurons to amyloid beta peptide, you get increased oxidative stress, lipid peroxidation, and there's some toxic products of lipid peroxidation that seem to do bad things for neurons. So then the ApoE, let's talk about, let's see, how should we do this? I want to try to merge that information
Yeah.
with what you're doing and with ApoE. There's been a huge amount of research on apolipoprotein E in relation to Alzheimer's because it can be a big risk factor. But the, the reason why is unclear. There's so many labs that have proposed so many different possibilities. So why don't let's talk about ApoE and then we'll come back to the
Sure.
lipid peroxidation and try to,
Put it all together.
yeah.
To solve Alzheimer's disease in a podcast.
Yeah, that'd
I
be
love
nice.
it. Yeah, so
But
ApoE,
we may end up coming up with some ideas what people can do to maybe
I love it. Yeah,
in their
sure.
lifestyle.
Yeah.
Okay.
So ApoE, I could tell you what I agree. People have been studying ApoE for decades. People have been studying ApoE and the three isoforms. So there's three major alleles in the population. ApoE3 is the most common. It's a neutral. ApoE4 is the risk factor, and ApoE2 is protective. So it'll actually protect you from the risk of neurodegeneration. And people have been studying why way longer than I've even been, studying ApoE or probably even doing
I've
research,
been
right?
alive.
Yeah. So, it's so well studied, and yet we don't have like a clear answer.
Yeah.
There are a ton of amazing studies that give clues along the way, and I think the the problem is that I don't think there is one answer. I think that it really it affects different cell types and different processes that are along the way. So what we focused on, looking at the ability to pull out those damaged oxidized lipids from neurons, is that E4 is very bad at it. Instead, it gets taken up and causes lysosomal dysfunction, whereas the protective isoform E2 and these other very rare variants that are also protective are better at pulling it out. So we think that that is one way, the ability to pull those toxic lipids out is one of several ways that can contribute to this risk factor.
And how did you go about showing that? So was this in cultured cells that have either E2, E3, E4, and then
Yeah.
yeah.
So we did it in two model systems. First is cell culture because that's our bread and butter, and that's what we do the majority of our experiments in. If you take neurons, cultured neurons, and you can do all sorts of stuff to them to stress them out or change their genotype. But we take these synthetic lipoprotein particles, so those POP-C, and then mix and match those isoforms and then look at how well they can pull out the lipids.
Oh,
So
I
you
see.
can look at that by imaging lipid storage. You can do lipidomics to look at what's
So
coming
you're
out.
actually adding adding the ApoEs to the culture medium?
Yep.
Huh,
Yeah.
okay. And
Yeah.
I don't know this. What's the distribution of ApoE in the brain? Is it mostly extracellular? Is it inside?
Oh, that's a good question. I'm not entirely sure about
Yeah, I'm
that.
not either.
Um,
But anyway,
I'm,
so I'm
I'm,
just wondering, is that
yeah.
adding it to the culture medium, is that where ApoE is really,
Well, that's where it's, every time I feel like I teach my undergrad something and I say something is fact, the next day I'll come out with a paper and I'll be like, oh, shoot, that's not 100% right. So ApoE definitely acts extracellularly. I would say up
yeah,
until
we have to be
maybe,
careful to use the
yeah,
word suggests or
yeah, yeah, that's right.
indicate
So
rather than shows or,
it certainly acts extracellularly. This is where the vast majority of its function and where it's been studied. However, I think it was maybe 3 or 4 years ago, Sarah Cohen's group from UNC Chapel Hill showed that ApoE actually escapes the secretory route and can act on lipid droplets directly, which is so
huh.
fascinating. So there's this whole other level of complexity to ApoE that outside of the
It kind of makes
secretory
sense
route.
from an evolutionary perspective, though. Why not, if this molecule is involved in movement, why not have it inside the cell as well
Yeah,
as outside?
yep, yep.
Yeah.
So it's
Because
definitely,
evolution tends to retain things that are good and give them additional duties, I guess
Yeah.
you would say.
Yeah. Yeah. Yeah.
Okay, and then
So we also did this in an ex vivo model. So what we do is we take intact hippocampi. So that's the region of your brain that's involved in learning and memory. It's one of the first places that starts to degenerate in Alzheimer's disease. And we could keep it alive. So it's rat, and you can keep this sort of chunk of tissue alive for several hours. So we got the idea from our collaborators who are electrophysiologists. They like this prep because you can stick electrodes in and look at communication without the input of the rest of the
we did
brain.
that some in my lab. I had an electrophysiologist. That's all he did.
Yeah. And I love this because you can add whatever drugs you want and you don't have to worry about like ethics, right? You're not going to hurt an animal or have
And
to worry about.
you can look at the activity in the circuits that
Yeah,
degenerate and also.
exactly.
Yeah.
So we took ApoE4 tissue. So this mimics
Uh,
disease. And we induced excitotoxicity. So that's that we forced the neurons to fire very, very rapidly, which is a form of stress.
did you
And
use an excitotoxin like canic acid, or how did you?
we used high potassium.
Okay. Yeah.
So they just start firing away. And if the E3s, so this mimics the healthy tissue, will just keep firing as long as the stimulus is present. The E4s will start firing, but then they'll sort of taper off and they can't maintain it. And we thought, well, they're probably dying because they're diseased tissue and they just can't withstand this type of stress. So then what we did was we took those protective particles and we bath applied it on top, and sure enough, they just keep firing. So we were
Wow.
able to rescue
Huh.
the neural activity or the ability to sustain that firing if we suck out those oxidized lipids and protect
And
them.
so you're adding E2 or E3?
We added E2 and E3 Christchurch. So Christchurch is one of those other protective variants that is gaining a lot of steam. The E3 was actually very robust in a lot of our assays as well.
Right. So okay, that's interesting.
Yeah.
No, my mind keeps like jumping all over the place, but now I'm wondering, what, um, yeah, so as far as therapy, so essentially you're showing E2 and the C3 Christchurch counteract
Yeah.
the adverse effect of E4, suggesting potentially some therapeutic approach where one introduces, or I guess somehow you can do this experimentally, but from a standpoint of people, uh, I guess the idea would be, people would get genotype. Do if I had a parent that had Alzheimer's,
Yeah.
then maybe I have E4, and then if I do have E4, then if I can somehow get E2,
Leverage
or there
this.
may be other ways to do the
Yeah.
same thing with
Sure.
drugs or something.
Yeah.
Yeah.
Ideally. So that's what we're
Okay.
hoping. I mean,
Now,
we work at a very fundamental level. So we
so
just,
now I'm gonna go back to the early 2000s.
okay.
And so we knew then that E4 was a risk factor and E2 was, and E3 were relatively protective. And so we just did simple thing. Okay, so what's the difference between E2, E3, and E4? It's in two amino acids,
Just do.
and those are E2, the amino acid in those two places is a cysteine. And E3, one of them is a cysteine. E4, neither of those two amino acids is a cysteine. So, we're thinking about working on oxidative stress. And one of the main endogenous antioxidants in cells is a three amino acid peptide called glutathione,
Yep.
and it has a cysteine. And that's why it's an antioxidant. So we had this simple-minded thing. We got recombinant E2, E3, and E4, and we'd previously shown that if we subject neurons to things that cause membrane lipid peroxidation, that is, attack of free radicals on certain lipids, the free radicals attack the double bond, and it causes a cleavage, a breaking of the lipid. And we do that with iron. Now, we didn't, back then we just call it iron-induced
Ugh,
cell toxicity. Now, someone had to come up with a
name.
sexy word for it. For now it's called ferroptosis. But anyway, so you get free radicals attack the lipids, you get release of several toxic lipids. One is 4-hydroxynonenal,
Mhm.
and it will bind to proteins on cysteine, lysine, or histidine residues. And glutathione is a sponge for this toxic aldehyde 4-hydroxynonenal. So we were studying that. We showed that this toxic aldehyde will impair the function of the glucose transporter in neurons, as well as interesting the sodium pump activity, and we found it increases vulnerability to excitotoxicity. So anyway, we just did this study. It wasn't that great. It was in like Journal of Neurochemistry, but essentially we showed that E2 sopped up more of this toxic aldehyde than E3 and E4. So we just had this simple-minded idea that E2 is protective because it's like sopping up
Yeah,
this toxic aldehyde. And so and we didn't study it much more after that.
I think it would be really interesting to see if we bath apply these exogenous particles, whether or not that's sucking it out, right? Like, does the cell
Yeah.
resolve that itself? Getting to know neurons a little more, I suspect they don't want to do that. They're busy. And so is
Um,
this a way to pull them out?
yeah. Yeah, I think you could come up with straightforward experiments to do
Yeah,
that. And 4-hydroxynonenal is really easy to work with, and
yeah,
you can just buy it from I don't know where now, Cayman or something.
probably.
I don't
They're
know.
great still.
I don't know if they're still in business.
Yes, they are. They have recently started working with us to help generate those lipoprotein particles, and
Oh, really?
yeah,
Good, good.
they're a terrific company.
Okay, let's now switch,
Yeah.
not switch, just extend it. So you told us what some of the different types of lipids are, most of them actually, and you mentioned glycolipids, and you had a very recently paper come out in Nature Cell Biology.
Yeah.
So you know, you publish everything in high impact journals.
Oh,
I had, we had some high impact papers, but I had so many people in the lab, and it's the kind of thing where they generate a lot of data. Maybe it's not something that's going to get in a top journal, but for their careers, it's important that
I don't think you
they're
need to.
publishing.
Yeah, I think I've published in a lot of low impact journals too. It's just what the story is, right?
Yep,
And I
yep.
think that all stories are.
And
Yeah.
it's very interesting if you go back, it may be changing now, but many Nobel Prize winners, if you go back
Oh yeah.
and look at, what did they publish 40, 50 years ago?
Journal of Biological Chemistry.
Yeah, yeah, yeah.
Yeah, for sure.
Okay, so this Nature Cell Biology paper, and I didn't read the whole paper, um, but it's very interesting. It's relevant to neurodegenerative disorders, in this case specifically Parkinson's disease, and you showed some interesting roles for lipids
Yeah.
potentially in Parkinson's disease. Go ahead and talk.
Yeah, sure. So, one of the major risk factors for Parkinson's disease is a mutation in a gene called GBA. And GBA encodes a lipid metabolism enzyme called GC, Ks, sorry. What this does, if you lost a function in this enzyme, is it accumulates a lipid called glucosylceramide. So in Parkinson's disease, you have increased glucosylceramide. And our question was, how does glucosylceramide affect Parkinson's disease pathology? What does this have to do with Parkinson's disease? And there's tons of papers, but our interests were on extracellular vesicles. And so the field for a really long time focuses on a subclass of extracellular vesicles because there's lots of different kinds of them that are called exosomes. And these are really small and they are within an endosome that fuse, that release. So that's one way you could get them out. But there's another class of extracellular vesicles called ectosomes or microvesicles that bud directly from the plasma membrane. So they just pinch off. And one of the big challenges in the literature that we saw is that it's very difficult to distinguish what type of vesicle you're working with once they're released. So people for a really long time just assumed everything was exosomes, so those little ones, because they were small or some markers. But we noted in one of the MICEV, the consortium of extracellular vesicle experts that were advising to exercise caution and trying to claim what they were without actually looking, right? So the only way to know if something is an ectosome, so budding from the surface, is to watch it happen, right? And so we're like, this sounds cool. We're a microscopy lab. We can do that. So a postdoc in the lab, excellent postdoc in the lab, Julie, devised a way to label the plasma membrane just with basic a GPI or a CAX or just a basic reporter and image the cells live. And what she realized is that when you have these mutations, these Parkinson's disease mutations that cause these increases in glucosylceramide, there's more ectosomes forming. So the neuron just sort of like is bubbling out these ectosomes. So that was the first thing that we thought was really interesting. And then she extended that work. So one of the disease mechanisms.
And will that occur like if you expose neurons to various types of stress, excitotoxic stress, oxidative stress, metabolic stress?
Yeah, so we haven't pushed too far on what types on a control background, all the different types of stressors that would phenocopy that. But we do see evidence of it happening in sort of the healthy control cells. It's just much less, right?
Okay.
So likely, yeah. So this could also extend to other neurodegenerative diseases too, right? You don't necessarily also glucosylceramide is a type of sphingolipid. There are all sorts of sphingolipid disturbances and all types of neurodegenerative diseases. It's not specific.
We did some work on that. I don't know if you know this. So I can't remember what year, it was early 2000s or something. We had a PNAS paper where there's a biologist in my lab who was really good at mass spec. And so he analyzed brain. He did essentially modified lipidomics, focusing on sphingolipids, ceramides of different sizes and so on. And in just normal brain aging in animals, in humans, and then in, what do we have? Alzheimer's, mild cognitive impairment, and controls. And then there was a guy, Norm Howie, who was a postdoc in the lab then, and there was Michelle Milkey, who's at Hopkins, and their group picked up on that, and they just ran with it. We never did anything more in my lab, but now there's all sorts of stuff going on with
well, people are validating that, right? With lots of increases in those sphingolipids like glucosylceramide in ApoE4 genetic backgrounds too.
They're using kind of biomarker, or not a biomarker. Yeah, kind of a biomarker approach.
Yeah, for disease that something's going wrong. maybe at early
Yeah.
stages, you
So,
can
so
pick
the
it up.
things you're doing, and I think a lot of the stuff we're doing are we're trying to understand mechanisms, get down to the nitty-gritty of what's actually happening that leads to degeneration of the neurons. So, aging's major risk factor, there's genetic risk factors, but they're all leading to kind of the same final outcome,
Yep.
degeneration of the same neurons, same clinical symptoms and stuff. And, so anyway, but go ahead with this.
Yeah. So in the Parkinson's disease story, one of the like characteristic phenotype of Parkinson's disease in terms of the pathophysiology is the way it spreads through the brain, right? It
Yeah.
takes a very specific spread from
The gut
yeah, it starts in the gut and then moves
through the
into
through the vagus nerve to the
the
brain
vagus
stem
nerve
and
to the brain stem. So by the time yeah, and then to the cortex. So at later
yeah.
stages, then you get that cognitive impairment. So we're sort of where you are along that spread dictates sort of the symptoms, right? Like most Parkinson's disease patients have GI symptoms before they have any.
Constipation, chronic
Yeah,
constipation.
exactly. And so one of the theories that's been proposed is that the misfolded protein alpha-synuclein, which is in Parkinson's disease, it's one of the major pathophysiological hallmarks, is that it's spreading from neuron to neuron in a prion-like fashion. So this misfolded protein goes to the next neuron and misfolds that protein that then goes to the next neuron and misfolds. And so one of the questions is, what are the ways that this protein can spread? So that's what we were interested in our ectosomes,
I
is
see.
that are these ectosomes filled with the pathogenic synuclein fibrils, and they are just loaded with it. There is so much of it in them.
Wow.
And it's a really cool imaging project because you can get fluorescently tagged alpha-synuclein fibrils and watch them form, and you can watch them then go to the next cells.
I should say this. I'm going to put in the description section, at least on the YouTube, yeah, I guess both for that and like Spotify, links to these papers
Yeah,
you're talking about because you have really beautiful,
thanks.
fantastic images where everything's very clear. It's like you're actually in the cell yourself.
Yeah.
Yeah.
I remember when Julie first came with the data when we finally got those fibrils, and like, you don't know what to expect, but we did not expect that much to be in them.
But do
It
you
was
think,
really
going back
strange.
to the previous things where you're looking at the neurons spitting out these oxidized lipids, do you think that this is a mechanism? So you're going to talk about this mechanism, potential mechanism for transneuronal propagation of the
Yeah.
synuclein pathology, but could the cells be spitting out the alpha-synuclein because they're recognizing they're under stress and they're trying to get rid of it?
I think that there's a lot of
And then eventually they can get rid of it.
interest.
Yeah.
Yeah, a lot of interest that this original like mechanism to rid themselves of these toxic proteins is initially beneficial for the neuron. And I think the same goes with the oxidized, the damaged lipids as well. Like this act of spewing these damaged toxic molecules out is initially protective, but then maybe disease follows. We need to track it, like who's picking it up and what's it doing to them, right? Like
Yeah.
glia can take up damaged lipids to a point.
Oh, wait a minute. So neurons communicate at synapses. Are
Yeah,
these ectosomes being released at synapses
they're all over the place.
all over the place? Okay.
All over the place, at least in vitro. We did a little bit in vivo. We tried two photon imaging live, which is a very challenging imaging experiment to do. And we have evidence of a vesicle that sort of buds from a neurite and then disappears from our field of view. So we can't, with two photon, we can't conclude a whole lot from that data alone, but at least it's consistent. But I think that would support that it can happen not just at synapse. I think it can happen at the cell body as
Okay,
well.
so with this genetic variant in the glucosylceramide, uh, yeah,
The GCAs. Yeah.
that causes the ectosome release, then, oh no, what was I going to say? Right. Oh, right. So does the alpha-synuclein stay in the vesicle?
Yeah.
And then does that vesicle fuse with another cell?
Gets taken up by more neurons.
Okay.
So it can transport.
Yeah.
And one of the phenotypes of misfolded synuclein is hyperphosphorylation. And you can take these vesicles and apply it to recipient neurons
Ah,
and show that the hyperphosphorylation of synuclein in the recipient neurons goes up. So it's a mechanism that can propagate synuclein pathology.
and did you do that in vivo? Did
No,
you,
that we did in vitro. In vivo, we used a drug to stimulate them, uh, and we found that there were more markers of the vesicles in recipient neurons. Yeah.
right, but can you isolate these vesicles from the culture medium and then kind of concentrate them and like inject them in the brain of, I guess you'd want
Yeah.
to do
Yeah.
wild type
Yeah.
mice and alpha-synuclein mutant mice, and then
See how they,
yeah.
or maybe differences in who takes it up.
And you said
And
you can have fluorescent synuclein.
yes. Yeah.
Yeah. So you
Yeah.
so who up there in Edmonton is there someone doing a lot of work in mouse models of Alzheimer's?
Yeah.
Yeah.
Yeah. We've got a
Okay.
handful of people up here with good animal models around. Yeah.
Okay.
Good people to work with.
Okay. Right. And then in the same study, you you looked at cells from human patients with, can you talk about that? So the
Yeah,
pluripotent
so
stem
a
cells.
lot of we toggled in that paper back and forth between two model systems. One is rat neurons because they're easier, more high throughput, and then for that we could use drugs to modulate GCA's activity or etc. But if the question was, is this really important in Parkinson's disease? So for that, we collaborated with Tom Durkin at McGill University. And got our hands on 5 different patient stem cell lines, each with their own mutation plus their isogenic corrected control. So that was 10 iPSC lines at one time, which if anyone's worked with iPSC lines, you can know how
And did you,
amazing my postdoc
so
was to
these
get
are
all this
just,
done.
a lot of people listening know what you're, this
Yeah,
are, but you take like fibroblasts from a
yeah,
patient and then you put, and this is the Japanese guy who got the Nobel Prize, Yamagata, or what's his name? I'm sorry, I can't remember his name. Anyway, you put a few genes in them and you can turn them into pluripotent stem cells that are capable of differentiating to any
yeah.
cell in the body or brain. So did you differentiate the cells into some neurons or
Yeah, so they were differentiated into dopaminergic neurons,
ah,
which is the type
yeah.
of neuron that is most affected in Parkinson's disease. And we didn't just have GBA mutations. We had GBA 3 lines with GBA mutations, different GBA mutations, and 2 lines with LRRK2 mutations. So LRRK2 is another risk factor for Parkinson's disease. It
That's a
also
causal. It's not a risk factor.
Right, that's
Yeah,
right.
because
And it also has reduced GCA's activity. So it has sort of the same phenotype that you have this enzyme
I
activity
see.
goes down and this glucosylceramide goes up. And so we were able to show more ectosomes forming on these 5 different Parkinson's patient backgrounds. It was,
Wow. Interesting.
yeah.
That's pretty convincing evidence. Um, okay. Are there ways, and I don't know, there probably are there ways to affect the amount of these lipids you've been talking about through diet or, yeah,
Yeah, I would say, you know, you always get asked these questions. I'm in a department of physiology, so
I don't know
sometimes,
the answer.
sometimes people, yeah, ask for like
Yeah, you have
lifestyle
to start with
advice.
the question, right?
I think, I think that the same answer applies to almost all diseases, right? If
Yeah,
you eat healthy diet rich in healthy fats,
yeah.
um, and you know, diets that allow for your body to handle cholesterol well, that doesn't necessarily mean don't eat any cholesterol. It just means that if you eat a balanced diet low in saturated animal fats
Yeah.
and general health, then your body can handle cholesterol. Your body needs cholesterol. um, and lifestyle choices will inevitably make their way throughout the tissues of your brain, including your brain, and that will reduce the risk, right? At the end of the day, a lot of these mutations are risk factors. They're not
And I think that,
guaranteed.
there's clearly in animal models, there's many, many different labs who have evidence that exercise, or we did a lot of work on intermittent fasting, can slow down anyway the progression of the pathology, the kinds of
Yep,
things you've been
yep,
talking about. So I think it's not unreasonable to predict that these interventions that are, and also I should say there's a lot of epidemiological evidence
yeah,
too that people who
yeah,
exercise regularly less likely to get Alzheimer's.
yep.
Uh, if you overeat and get insulin resistance, you're more likely. So it kind of makes sense that these lifestyle factors would affect the processes you've been studying in a good way.
Absolutely.
And so then the question is, how do you study this? I don't have a lab anymore. So, I mean, you have to have ways to look at this in vivo, I guess. What about the cerebrospinal fluid?
Yeah,
Uh, the fluid so these are being released from cells. So probably these vesicles, like in a alpha-synuclein in mice, and this can be done, I mean, they're small, but you can get cerebrospinal fluid from them. Would they have a lot of these ectosome
yeah,
size vesicles loaded with alpha-synuclein? And then you could just put running wheels in the cage or do, you know, every other day food deprivation,
yeah.
fasting, and then just take this CSF, see the, I don't know, there are some experiments you can do.
I think there's a lot there. I think that one right now we're very hyper focused on this neuron to glia transport, and I think that that is
Um,
limited in how
you've,
protective that can
oh,
be.
wait a minute. Yeah. Oh, you haven't got tenure yet.
Uh Technically.
Okay. Yeah. Once you get tenure, you're not going to have to worry about what you do. Just do what you want to do.
I love that.
That's what I did.
It's more fun.
Wherever, but you do have, you have grants. You're funded by grants, and your grant
Yeah,
has, specific aims. These are the goals of the grant. And if you're going to try to continue and extend that grant for more years, then you have
you
to
gotta
show
have some deliverables.
you have to have some deliverables. But what I did,
There's room to
you're talking about your mentor
McGill,
from
Peter.
McGill, yeah.
Yeah,
Uh, so one thing I did, I would get a grant, and then I had a lot of postdocs. I mean, just less effort by myself and more productive. That's just the way it is. So then there were certain opportunities for postdocs to get their own funding uh, through the NIH or private foundations like
Yeah.
Alzheimer's Association or whatever. So I would, after the postdoc was in the lab for couple of years, I'd have them apply for their own funding. Then if they got their own funding, then that freed up the money from my grant for a postdoc, and I'd bring in another postdoc. So
Yeah.
by the end of 5-year grants, I'd have 3 postdocs. And then I could kind of, have the one postdoc focus on the aims of the grant and the others
Someone,
start to do new things.
yeah.
So
I think it's the only way because in order to get that next grant, you need preliminary data, but you
yeah.
don't have money to
But then
generate
you
that
can get
preliminary.
another grant for another,
Yeah.
it's something a little different than what you've been
Yeah.
doing.
Yeah.
So I don't know. Yeah.
There's definitely room to play, which I think
Yeah.
is one of the draws to academic science
Yeah. Yeah.
over industry where there's more focused goals. Here, if I have some crazy idea and I want to start exploring it, there is room to pursue that.
Yeah.
I like that about
Yeah.
this
And
job.
Canada is not as dumb as us. It's well, not me personally, but government. It's really crazy, defunding
Yeah,
scientific research.
it's really scary.
That's what got our country where we are, science and technology.
Yeah,
And
at the forefront or
and
the
long and
leader.
long and long thinking, investing in long-term initiatives and projects. And
Yeah, it's
it's
definitely short-sighted, and I think it will hurt the U.S. long term. For us, hopefully, we can
you guys
maintain.
up there, you're keeping your elbows up
We're
and
trying. Yeah. We're also recruiting from the US, right? There's this new
that's
impact grant
there's
right now that is trying
this is
to
something, and
pull
not
people.
just with this recent administration,
Yeah,
although it's really dramatic now, but there has been kind of a slow brain drain. And the first thing I saw was, I've had a lot of postdocs from China over the
yeah,
35 years or whatever. And it used to be they would never think about going back to China.
yeah,
They would get some position in the United States.
yeah,
And then definitely within the last 10 years or so, many of these postdocs are going back to China because China has really good funding for research. They give them really nice lab startup
yeah.
and fundings to get them going. And we can already see the results. China is getting
It's noticeable.
way ahead.
That's
They're
right.
just accelerating,
Yeah,
and we're being stupid. Uh,
yeah. You're
it's,
dropping off.
yeah,
We're trying to hold steady,
yeah,
but we are. Um, I am noticing more applicants to to my lab or to our departments that would normally go to the US.
yeah,
Also in training my own trainees when they go to move on to their postdocs, for instance, um, early talks were like thinking about places to go in the US, and now it's Europe. Everybody's going to Europe
yeah,
if they want to leave Canada. Either they'll stay in Canada and move around, or if they want to get some international experience, then they're looking towards Europe instead of the US.
yeah.
So we definitely see it here too.
It's a tough time for people who just got their PhD as
Yeah,
well as postdocs in the United States. Yeah.
yeah.
Okay, now I don't want to end on that. What are we going to end on?
Something more optimistic about the future.
Well, what you're doing gives us optimism,
I
what you
hope so.
talked about. So I guess I do have a last question.
Okay.
I guess we kind of touched on it, but uh, how might your research lead to new ways for preventing and treating neurological disorders? What are you thinking?
I think ways that we can help neurons clear misfolded protein or damaged lipids. So a lot of people study misfolded proteins. We can focus on the damaged lipids because I think that that's less
Yeah,
well tapped.
yeah,
Um, And whether that's getting it out of the neurons, I think we need to be really careful about who we're shipping those damaged lipids to. If it's just shifting the burden to the glia, I think that isn't necessarily the solution. Maybe there's other, maybe
Enhancing the removal inside the cell
yeah,
too.
and it might be out of the brain entirely. It might be through the CSF. It might be glymphatic, right? There's a
Yeah.
lot of stuff here that's not touched. Or, there was a lot of reasonable studies looking at antioxidants, and antioxidants didn't cure Alzheimer's disease. However, a lot of antioxidants that were being looked at were not lipid soluble antioxidants, right? They don't solve this particular kind of reactive oxygen species. Yeah. And maybe to revisit that, maybe that's not going to be a cure, but maybe that combined with something else can help slow down progression and have a little more efficacy than some of the earlier antioxidant studies that we've tried before.
Yeah, I think that the oral anti, you go to vitamin or supplement store,
Yeah,
everything says
that's not
antioxidant.
good.
But what I think we need to figure out, and what you're doing, yeah, is how can we stimulate cells, neurons, in ways that they beef up or enhance their endogenous
Yeah.
defenses against oxidation.
GPX4, more GPX4.
Yeah, or more stimulate antioxidant
Yeah.
enzymes, DNA repair enzymes, glutathione,
Yep.
whatever. Help the cells help themselves.
Yeah.
And that's what we're doing with exercise, with
Yep.
intermittent fasting, or keeping your brain active.
Yeah.
And I'm writing a third book. So I've written two books since I retired, but the third one, the working title, and it's actually going to be a Canadian publisher,
Okay.
University of Toronto
Terrific.
Press, but it's called tentatively Challenge, Recover, Repeat. And it's about the science of what happens to cells during exercise, fasting, exercising our neurons, and then what happens during recovery, resting, eating, sleeping, and how cycling between the mild physiological stress or challenge and recovery over time can make cells stronger and more resilient. So,
Yeah.
yeah.
Yep. I think you're onto something there. I think that'll be a great topic to write
So I
about.
think, yeah, but I think you can incorporate some of that into your work, that same
Yeah.
idea. Use good stresses.
Yeah.
Yeah.
Cool.
Okay, Maria, it's great to meet you and talk to you, and
Yeah.
it's great to see young investigators like you doing such really amazing work.
Thanks.
Makes me want to get back in the lab. But okay, thanks, and have good
Thank you so much for having me.
spring's on its way, I think, even in Canada.
Eventually.
Yeah.
Okay.
All right.
Thank you.
Bye.